Left photo shows a long, straight highway in the middle of a desert. Right photo shows a mouse. Figure 46. 1. In the southwestern United States, rainy weather causes an increase in production of pinyon nuts, causing the deer mouse population to explode. Deer mice may carry a virus called sin nombre, a hantavirus, that causes respiratory disease in humans and has a high fatality rate. In 1992-1993, wet El Niño weather caused a sin nombre epidemic. Navajo healers who were aware of the link between this disease and weather predicted the outbreak. Credit highway, modification of work by Philip Capper. Credit mouse, modification of work by USFWS. Chapter outline. 46. 1. Ecology of ecosystems. 46. 2. Energy flow through ecosystems. 46. 3. Biogeochemical cycles. In 1993, an interesting example of ecosystem dynamics occurred when a rare lung disease struck inhabitants of the southwestern United States. This disease had an alarming rate of fatalities, killing more than half of early patients, many of whom were Native Americans. These formerly healthy young adults died from complete respiratory failure. The disease was unknown, and the Centers for Disease Control, CDC, the United States government agency responsible for managing potential epidemics, was brought in to investigate. The scientists could have learned about the disease had they known to talk with the Navajo healers who lived in the area and who had observed the connection between rainfall and mice populations, thereby predicting the 1993 outbreak. The cause of the disease, determined within a few weeks by the CDC investigators, was the hantavirus known as sin nombre, the virus with no name. With insights from traditional Navajo medicine, scientists were able to characterize the disease rapidly and institute effective health measures to prevent its spread. This example illustrates the importance of understanding the complexities of ecosystems and how they respond to changes in the environment. Learning objectives. By the end of this section, you will be able to do the following. Describe the basic ecosystem types. Explain the methods that ecologists use to study ecosystem structure and dynamics. Identify the different methods of ecosystem modeling. Differentiate between food chains and food webs, and recognize the importance of each. Life in an ecosystem is often about competition for limited resources, a characteristic of the theory of natural selection. Competition in communities, all living things within specific habitats, is observed both within species and among different species. The resources for which organisms compete include organic material, sunlight, and mineral nutrients, which provide the energy for living processes and the matter to make up organisms' physical structures. Other critical factors influencing community dynamics are the components of its physical and geographic environment. A habitat's latitude, amount of rainfall, topography, elevation, and available species. These are all important environmental variables that determine which organisms can exist within a particular area. An ecosystem is a community of living organisms and their interactions with their abiotic, non-living environment. Ecosystems can be small, such as the tide pools found near the rocky shores of many oceans, or large, such as the Amazon rainforest in Brazil. Figure 46.2. Left photo shows a rocky tide pool with seaweed and snails. Right photo shows the Amazon rainforest landscape, with seemingly every inch of land covered by plants of some kind. Figure 46. 2a, a tidal pool ecosystem in Matinicus Island in Maine, is a small ecosystem, while the B, Amazon rainforest in Brazil, is a large ecosystem. Credit A, modification of work by Takamabiblot/Flickr. Credit B, modification of work by Ivan M. Linaric. There are three broad categories of ecosystems based on their general environment: freshwater, ocean water, and terrestrial. Within these broad categories are individual ecosystem types based on the organisms present and the type of environmental habitat. Ocean ecosystems are the most common, comprising over 70% of the Earth's surface and consisting of three basic types: shallow ocean, deep ocean water, and deep ocean surfaces, the low-depth areas of the deep oceans. The shallow ocean ecosystems include extremely biodiverse coral reef ecosystems, and the deep ocean surface is known for its large numbers of plankton and krill, small crustaceans, that support it. These two environments are especially important to aerobic respirators worldwide, as the phytoplankton perform 40% of all photosynthesis on Earth. Although not as diverse as the other two, deep ocean ecosystems contain a wide variety of marine organisms. Such ecosystems exist even at the bottom of the ocean, where light is unable to penetrate through the water. Freshwater ecosystems are the rarest, occurring on only 1/8% of the Earth's surface. Lakes, rivers, streams, and springs comprise these systems. They are quite diverse, and they support a variety of fish, amphibians, reptiles, insects, phytoplankton, fungi, and bacteria. Terrestrial ecosystems, also known for their diversity, are grouped into large categories called biomes, such as tropical rainforests, savannas, deserts, coniferous forests, deciduous forests, and tundra. Grouping these ecosystems into just a few biome categories obscures the great diversity of the individual ecosystems within them. For example, there is great variation in desert vegetation. The saguaro cacti and other plant life in the Sonoran Desert in the United States are relatively abundant compared to the desolate rocky desert of Boa Vista, an island off the coast of Western Africa. Figure 46.3. Photo A shows saguaro cacti that look like telephone poles with arms extended from them. Photo B shows a barren plain of red soil littered with rocks. Figure 46. 3 desert ecosystems, like all ecosystems, can vary greatly. The desert in A, Saguaro National Park, Arizona, has abundant plant life, while the rocky desert of B, Boa Vista Island, Cape Verde, Africa, is devoid of plant life. Credit A, modification of work by J. Galvin. Credit B, modification of work by Ingo Whirlburn. Ecosystems are complex, with many interacting parts. They are routinely exposed to various disturbances or changes in the environment that affect their compositions. Yearly variations in rainfall and temperature, and the slower processes of plant growth, which may take several years. Many of these disturbances result from natural processes. For example, when lightning causes a forest fire and destroys part of a forest ecosystem, the ground is eventually populated by grasses, then by bushes and shrubs, and later by mature trees, restoring the forest to its former state. The impact of environmental disturbances caused by human activities is as important as the changes wrought by natural processes. Human agricultural practices, air pollution, acid rain, global deforestation, overfishing, eutrophication, oil spills, and waste dumping on land and into the ocean are all issues of concern to conservationists. Equilibrium is the steady state of an ecosystem where all organisms are in balance with their environment and with each other. In ecology, 2 parameters are used to measure changes in ecosystems: resistance and resilience. Resistance is the ability of an ecosystem to remain at equilibrium in spite of disturbances. Resilience is the speed at which an ecosystem recovers equilibrium after being disturbed. Ecosystem resistance and resilience are especially important when considering human impact. The nature of an ecosystem may change to such a degree that it can lose its resilience entirely. This process can lead to the complete destruction or irreversible altering of the ecosystem. Food chains and food webs. The term food chain is sometimes used metaphorically to describe human social situations. Individuals who are considered successful are seen as being at the top of the food chain, consuming all others for their benefit, whereas the less successful are seen as being at the bottom. The scientific understanding of a food chain is more precise than in its everyday usage. In ecology, a food chain is a linear sequence of organisms through which nutrients and energy pass. Primary producers, primary consumers, and higher-level consumers are used to describe ecosystem structure and dynamics. There is a single path through the chain. Each organism in a food chain occupies what is called a trophic level. Depending on their role as producers or consumers, species or groups of species can be assigned to various trophic levels. In many ecosystems, the bottom of the food chain consists of photosynthetic organisms, plants and/or phytoplankton, which are called primary producers. The organisms that consume the primary producers are herbivores, the primary consumers. Secondary consumers are usually carnivores that eat the primary consumers. Tertiary consumers are carnivores that eat other carnivores. Higher-level consumers feed on the next lower trophic levels, and so on, up to the organisms at the top of the food chain, the apex consumers. In the Lake Ontario food chain shown in Figure 46. 4. The Chinook salmon is the apex consumer at the top of this food chain. In this illustration, the bottom trophic level is the primary producer, which is green algae. The primary consumers are mollusks or snails. The secondary consumers are small fish called slimy sculpin. The tertiary and apex consumer is Chinook salmon. Figure 46. For these are the trophic levels of a food chain in Lake Ontario at the United States-Canada border. Energy and nutrients flow from photosynthetic green algae at the bottom to the top of the food chain, the Chinook salmon. One major factor that limits the length of food chains is energy. Energy is lost as heat between each trophic level due to the second law of thermodynamics. Thus, after a limited number of trophic energy transfers, the amount of energy remaining in the food chain may not be great enough to support viable populations at yet a higher trophic level. The loss of energy between trophic levels is illustrated by the pioneering studies of Howard T. Odom in the Silver Springs, Florida, ecosystem in the 1940s, Figure 46. 5. The primary producers generated 20,819 kcal/m2/, or kilocalories per square meter per year. The primary consumers generated 3,368 kcal/m2/, or the secondary consumers generated 383 kcal/m2/, or, and the tertiary consumers only generated 21 kcal/m2/, or. Thus, there is little energy remaining for another level of consumers in this ecosystem. Graph shows energy content in different trophic levels. The energy content of primary producers is over 20,000 kilocalories per meter squared per year. The energy content of primary consumers is much smaller, about 3,400 kilocalories per meter squared per year. The energy content of secondary consumers is 383 kilocalories per meter squared per year, and the energy content of tertiary consumers is only 21 kilocalories per meter squared per year. Figure 46. 5. The relative energy and trophic levels in a Silver Springs, Florida, ecosystem is shown. Each trophic level has less energy available and supports fewer organisms at the next level. There is one problem when using food chains to accurately describe most ecosystems. Even when all organisms are grouped into appropriate trophic levels, some of these organisms can feed on species from more than one trophic level. Likewise, some of these organisms can be eaten by species from multiple trophic levels. In other words, the linear model of ecosystems, the food chain, is not completely descriptive of ecosystem structure. A holistic model, which accounts for all the interactions between different species and their complex interconnected relationships with each other and with the environment, is a more accurate and descriptive model for ecosystems. A food web is a graphic representation of a holistic, nonlinear web of primary producers, primary consumers, and higher-level consumers used to describe ecosystem structure and dynamics, Figure 46. 6. The bottom level of the illustration shows primary producers, which include diatoms, green algae, blue-green algae, and flagellates. The next level includes the primary consumers that eat primary producers. These include invertebrates such as waterflias and mussels. Primary consumers are in turn eaten by secondary consumers, which are typically small fish and shrimp. The small fish are eaten by larger fish, the tertiary or apex consumers. Finally, all fish are eaten by the sea lamprey. For the most part, every organism at a given level eats every organism in the level below and nothing else, but there are exceptions. For example, not all secondary consumers eat all primary consumers. Lake whitefish only eat zebra and quagga mussels, round goby eat all primary consumers, and the other secondary consumers eat every primary consumer except for the zebra and quagga mussels. The secondary consumer yellow perch eats two other secondary consumers from its own level, the slimy sculpin and the rainbow smelt, in addition to eating primary consumers from the level below. Some organisms can also skip a level. The secondary consumer opossum, shrimp, eats most primary consumers in the level immediately below and all of the primary producers 2 levels below. Sea lamprey eat all tertiary and secondary consumers. Thus, the food web is complex with interwoven layers. Figure 46. 6. This food web shows the interactions between organisms across trophic levels in the Lake Ontario ecosystem. Primary producers are outlined in green, primary consumers in orange, secondary consumers in blue, and tertiary apex, consumers in purple. Arrows point from an organism that is consumed to the organism that consumes it. Notice how some lines point to more than one trophic level. For example, the opossum shrimp eats both primary producers and primary consumers. Credit NOAA-GLERL. A comparison of the two types of structural ecosystem models shows strength in both. Food chains are more flexible for analytical modeling, are easier to follow, and are easier to experiment with, whereas food web models more accurately represent ecosystem structure and dynamics, and data can be directly used as input for simulation modeling. Link to learning. Head to this online interactive simulator to investigate food web function. In the Interactive Labs box, under Underline Food Web and Underline, click Step 1. Read the instructions first, and then click Step 2 for additional instructions. When you are ready to create a simulation, in the upper right corner of the Interactive Labs box, click Open Simulator. Two general types of food webs are often shown interacting within a single ecosystem. A grazing food web, such as the Lake Ontario food web in Figure 46. 6 has plants or other photosynthetic organisms at its base, followed by herbivores and various carnivores. A detrital food web consists of a base of organisms that feed on decaying organic matter, dead organisms, called decomposers or detritivores. These organisms are usually bacteria or fungi that recycle organic material back into the biotic part of the ecosystem, as they themselves are consumed by other organisms. As all ecosystems require a method to recycle material from dead organisms, most grazing food webs have an associated detrital food web. For example, in a meadow ecosystem, plants may support a grazing food web of different organisms, primary and other levels of consumers, while at the same time supporting a detrital food web of bacteria, fungi, and detrivorous invertebrates feeding off dead plants and animals. Evolution connection. Three-spine stickleback. It is well established by the theory of natural selection that changes in the environment play a major role in the evolution of species within an ecosystem. However, little is known about how the evolution of species within an ecosystem can alter the ecosystem environment. In 2009, Dr. Luke Harmon, from the University of Idaho, published a paper that for the first time showed that the evolution of organisms into subspecies can have direct effects on their ecosystem environment. 1. The three-spined stickleback, Gasterosteus achillidas, is a freshwater fish that evolved from a saltwater fish to live in freshwater lakes about 10,000 years ago, which is considered a recent development in evolutionary time. Figure 46. 7. Over the last 10,000 years, these freshwater fish then became isolated from each other in different lakes. Depending on which lake population was studied, findings showed that these sticklebacks then either remained as one species or evolved into two species. The divergence of species was made possible by their use of different areas of the pond for feeding called micro-niches. Dr. Harmon and his team created artificial pond microcosms in 250-gallon tanks and added muck from freshwater ponds as a source of zooplankton and other invertebrates to sustain the fish. In different experimental tanks, they introduced one species of stickleback from either a single species or double species lake. Over time, the team observed that some of the tanks bloomed with algae while others did not. This puzzled the scientists, and they decided to measure the water's dissolved organic carbon, DOC, which consists of mostly large molecules of decaying organic matter that give pond water its slightly brownish color. It turned out that the water from the tanks with two species fish contained larger particles of DOC, and hence darker water, than water with single species fish. This increase in DOC blocked the sunlight and prevented algal blooming. Conversely, the water from the single species tank contained smaller DOC particles, allowing more sunlight penetration to fuel the algal blooms. This change in the environment, which is due to the different feeding habits of the stickleback species in each lake type, probably has a great impact on the survival of other species in these ecosystems, especially other photosynthetic organisms. Thus, the study shows that, at least in these ecosystems, the environment and the evolution of populations have reciprocal effects that may now be factored into simulation models. Photo shows two small fish swimming above a rocky bottom. Figure 46. 7. The three-spined stickleback evolved from a saltwater fish to freshwater fish. Credit, Barrett Paul, USFWS. Research into ecosystem dynamics, ecosystem experimentation, and modeling. The study of the changes in ecosystem structure caused by changes in the environment, disturbances, or by internal forces is called ecosystem dynamics. Ecosystems are characterized using a variety of research methodologies. Some ecologists study ecosystems using controlled experimental systems, while some study entire ecosystems in their natural state, and others use both approaches. A holistic ecosystem model attempts to quantify the composition, interaction, and dynamics of entire ecosystems. It is the most representative of the ecosystem in its natural state. A food web is an example of a holistic ecosystem model. However, this type of study is limited by time and expense, as well as the fact that it is neither feasible nor ethical to do experiments on large natural ecosystems. It is difficult to quantify all different species in an ecosystem and the dynamics in their habitat, especially when studying large habitats such as the Amazon rainforest. For these reasons, scientists study ecosystems under more controlled conditions. Experimental systems usually involve either partitioning a part of a natural ecosystem that can be used for experiments, termed a mesocosm, or by recreating an ecosystem entirely in an indoor or outdoor laboratory environment, which is referred to as a microcosm. A major limitation to these approaches is that removing individual organisms from their natural ecosystem, or altering a natural ecosystem, through partitioning may change the dynamics of the ecosystem. These changes are often due to differences in species numbers and diversity, and also to environment alterations caused by partitioning, mesocosm, or recreating microcosm, the natural habitat. Thus, these types of experiments are not totally predictive of changes that would occur in the ecosystem from which they were gathered. As both of these approaches have their limitations, some ecologists suggest that results from these experimental systems should be used only in conjunction with holistic ecosystem studies to obtain the most representative data about ecosystem structure, function, and dynamics. Scientists use the data generated by these experimental studies to develop ecosystem models that demonstrate the structure and dynamics of ecosystems. They use three basic types of ecosystem modeling in research and ecosystem management: a conceptual model, an analytical model, and a simulation model. A conceptual model is an ecosystem model that consists of flowcharts to show interactions of different compartments of the living and non-living components of the ecosystem. A conceptual model describes ecosystem structure and dynamics and shows how environmental disturbances affect the ecosystem. However, its ability to predict the effects of these disturbances is limited. Analytical and simulation models, in contrast, are mathematical methods of describing ecosystems that are indeed capable of predicting the effects of potential environmental changes without direct experimentation, although with some limitations as to accuracy. An analytical model is an ecosystem model that is created using simple mathematical formulas to predict the effects of environmental disturbances on ecosystem structure and dynamics. A simulation model is an ecosystem model that is created using complex computer algorithms to holistically model ecosystems and to predict the effects of environmental disturbances on ecosystem structure and dynamics. Ideally, these models are accurate enough to determine which components of the ecosystem are particularly sensitive to disturbances, and they can serve as a guide to ecosystem managers, such as conservation ecologists or fisheries biologists, in the practical maintenance of ecosystem health. Conceptual models. Conceptual models are useful for describing ecosystem structure and dynamics, and for demonstrating the relationships between different organisms in a community and their environment. Conceptual models are usually depicted graphically as flow charts. The organisms and their resources are grouped into specific compartments, with arrows showing the relationship and transfer of energy or nutrients between them. Thus, these diagrams are sometimes called compartment models. To model the cycling of mineral nutrients, organic and inorganic nutrients are subdivided into those that are bioavailable, ready to be incorporated into biological macromolecules, and those that are not. For example, in a terrestrial ecosystem near a deposit of coal, carbon will be available to the plants of this ecosystem as carbon dioxide gas in a short-term period, not from the carbon-rich coal itself. However, over a longer period, microorganisms capable of digesting coal will incorporate its carbon or release it as natural gas, methane, CH4, changing this unavailable organic source into an available one. This conversion is greatly accelerated by the combustion of fossil fuels by humans, which releases large amounts of carbon dioxide into the atmosphere. This is thought to be a major factor in the rise of the atmospheric carbon dioxide levels in the industrial age. The carbon dioxide released from burning fossil fuels is produced faster than photosynthetic organisms can use it. This process is intensified by the reduction of photosynthetic trees because of worldwide deforestation. Most scientists agree that high atmospheric carbon dioxide is a major cause of global climate change. Conceptual models are also used to show the flow of energy through particular ecosystems. Figure 46.8 is based on Howard T. Odom's classical study of the Silver Springs, Florida, holistic ecosystem in the mid-20th century. 2. This study shows the energy content and transfer between various ecosystem compartments. Visual connection. Flow chart shows that the ecosystem absorbs 170,000 calories per meter squared per year of sunlight. Primary producers have a gross productivity of 20,810 calories per meter squared per year. 13,187 calories per meter squared per year is lost to respiration and heat. So the net productivity of primary producers is 7,623 calories per meter squared per year. 4,250 calories per meter squared per year is passed on to decomposers, and the remaining 3,373 calories per meter squared per year is passed on to primary consumers. Thus, the gross productivity of primary consumers is 3,373 calories per meter squared per year. 2,270 calories per meter squared per year is lost to heat and respiration, resulting in a net productivity for primary consumers of 1,103 calories per meter squared per year. 720 calories per meter squared per year is lost to decomposers, and 383 calories per meter squared per year becomes the gross productivity of secondary consumers. 272 calories per meter squared per year is lost to heat and respiration. So the net productivity for secondary consumers is 111 calories per meter squared per year. 90 calories per meter squared per year is lost to decomposers, and the remaining 21 calories per meter squared per year becomes the gross productivity of tertiary consumers. 16 calories per meter squared per year is lost to respiration and heat, so the net productivity of tertiary consumers is 5 calories per meter squared per year. All this energy is lost to decomposers. In total, decomposers use 5,065 calories per meter squared per year of energy, and 20,810 calories per meter squared per year is lost to respiration and heat. Figure 46.8. This conceptual model shows the flow of energy through a spring ecosystem in Silver Springs, Florida. Notice that the energy decreases with each increase in trophic level. Why do you think the value for gross productivity of the primary producers is the same as the value for total heat and respiration? 20,810 kcal/m2/er. Analytical and simulation models. The major limitation of conceptual models is their inability to predict the consequences of changes in ecosystem species and/or environment. Ecosystems are dynamic entities and subject to a variety of abiotic and biotic disturbances caused by natural forces and/or human activity. Ecosystems altered from their initial equilibrium state can often recover from such disturbances and return to a state of equilibrium. As most ecosystems are subject to periodic disturbances and are often in a state of change, they are usually either moving toward or away from their equilibrium state. There are many of these equilibrium states among the various components of an ecosystem, which affects the ecosystem overall. Furthermore, as humans have the ability to greatly and rapidly alter the species content and habitat of an ecosystem, the need for predictive models that enable understanding of how ecosystems respond to these changes becomes more crucial. Analytical models often use simple, linear components of ecosystems, such as food chains, and are known to be complex mathematically. Therefore, they require a significant amount of mathematical knowledge and expertise. Although analytical models have great potential, their simplification of complex ecosystems is thought to limit their accuracy. Simulation models that use computer programs are better able to deal with the complexities of ecosystem structure. A recent development in simulation modeling uses supercomputers to create and run individual-based simulations, which accounts for the behavior of individual organisms and their effects on the ecosystem as a whole. These simulations are considered to be the most accurate and predictive of the complex responses of ecosystems to disturbances. Link to learning. Visit the Darwin Project to view a variety of ecosystem models, including simulations that model predator-prey relationships to learn more. Learning objectives. By the end of this section, you will be able to do the following. Describe how organisms acquire energy in a food web and in associated food chains. Explain how the efficiency of energy transfers between trophic levels affects ecosystem structure and dynamics. Discuss trophic levels and how ecological pyramids are used to model them. All living things require energy in one form or another. Energy is required by most complex metabolic pathways, often in the form of adenosine triphosphate, ATP, especially those responsible for building large molecules from smaller compounds, and life itself is an energy-driven process. Living organisms would not be able to assemble macromolecules, proteins, lipids, nucleic acids, and complex carbohydrates from their monomeric subunits without a constant energy input. It is important to understand how organisms acquire energy and how that energy is passed from one organism to another through food webs and their constituent food chains. Food webs illustrate how energy flows directionally through ecosystems, including how efficiently organisms acquire it, use it, and how much remains for use by other organisms of the food web. How organisms acquire energy in a food web. Energy is acquired by living things in three ways: photosynthesis, chemosynthesis, and the consumption and digestion of other living or previously living organisms by heterotrophs. Photosynthetic and chemosynthetic organisms are both grouped into a category known as autotrophs. Organisms capable of synthesizing their own food, more specifically, capable of using inorganic carbon as a carbon source. Photosynthetic autotrophs, photoautotrophs, use sunlight as an energy source, whereas chemosynthetic autotrophs, chemoautotrophs, use inorganic molecules as an energy source. Autotrophs are critical for all ecosystems. Without these organisms, energy would not be available to other living organisms and life itself would not be possible. Photoautotrophs, such as plants, algae, and photosynthetic bacteria, serve as the energy source for a majority of the world's ecosystems. These ecosystems are often described by grazing food webs. Photoautotrophs harness the solar energy of the sun by converting it to chemical energy in the form of ATP and NADP. The energy stored in ATP is used to synthesize complex organic molecules, such as glucose. Chemoautotrophs are primarily bacteria that are found in rare ecosystems where sunlight is not available, such as in those associated with dark caves or hydrothermal vents at the bottom of the ocean. Figure 46. 9. Many chemoautotrophs in hydrothermal vents use hydrogen sulfide H2S, which is released from the vents as a source of chemical energy. This allows chemoautotrophs to synthesize complex organic molecules, such as glucose, for their own energy and in turn supplies energy to the rest of the ecosystem. Photo shows shrimp, lobster, and white crabs crawling on a rocky ocean floor littered with mussels. Figure 46. 9 swimming shrimp, a few squat lobsters, and hundreds of vent mussels are seen at a hydrothermal vent at the bottom of the ocean. As no sunlight penetrates to this depth, the ecosystem is supported by chemoautotrophic bacteria and organic material that sinks from the ocean's surface. This picture was taken in 2006 at the submerged NW Eifuku volcano off the coast of Japan by the National Oceanic and Atmospheric Administration, NOAA. The summit of this highly active volcano lies 1535 m below the surface. Productivity within trophic levels. Productivity within an ecosystem can be defined as the percentage of energy entering the ecosystem incorporated into biomass in a particular trophic level. Biomass is the total mass, in a unit area at the time of measurement, of living or previously living organisms within a trophic level. Ecosystems have characteristic amounts of biomass at each trophic level. For example, in the English Channel ecosystem, the primary producers account for a biomass of 4 gm2 grams per square meter, while the primary consumers exhibit a biomass of 21 gm2. The productivity of the primary producers is especially important in any ecosystem because these organisms bring energy to other living organisms by photoautotrophy or chemoautotrophy. The rate at which photosynthetic primary producers incorporate energy from the sun is called gross primary productivity. An example of gross primary productivity is shown in the compartment diagram of energy flow within the Silver Springs aquatic ecosystem, as shown, figure 46. 8. In this ecosystem, the total energy accumulated by the primary producers, gross primary productivity, was shown to be 20,810 kcal/m2/. Because all organisms need to use some of this energy for their own functions, like respiration and resulting metabolic heat loss, scientists often refer to the net primary productivity of an ecosystem. Net primary productivity is the energy that remains in the primary producers after accounting for the organism's respiration and heat loss. The net productivity is then available to the primary consumers at the next trophic level. In our Silver Springs example, 13,187 of the 20,810 kcal/m2/ were used for respiration or were lost as heat, leaving 7,633 kcal/m2/ of energy for use by the primary consumers. Ecological efficiency, the transfer of energy between trophic levels, as illustrated in figure 46. 8. As energy flows from primary producers through the various trophic levels, the ecosystem loses large amounts of energy. The main reason for this loss is the second law of thermodynamics, which states that whenever energy is converted from one form to another, there is a tendency toward disorder entropy in the system. In biologic systems, this energy takes the form of metabolic heat, which is lost when the organisms consume other organisms. In the Silver Springs ecosystem example, figure 46. 8, we see that the primary consumers produced 1103 kcal/m2/, or from the 3373 kcal/m2/, or of energy available to them from the primary producers. The primary consumers used 3373 kcal/m2/, or from the 7618 kcal/m2/, or produced by the primary producers, as 4250 kcal/m2/, or goes to the decomposers. The measurement of energy transfer efficiency between two successive trophic levels is termed the trophic level transfer efficiency, TLTA, and is defined by the formula. TLTA, production at present trophic level, production at previous trophic level, times 100. In Silver Springs, the TLTA between the first two trophic levels was approximately 48%. The low efficiency of energy transfer between trophic levels is usually the major factor that limits the length of food chains observed in a food web. The fact is, after 4 to 6 energy transfers, there is not enough energy left to support another trophic level. In the Lake Ontario example, shown in figure 46, only 3 energy transfers occurred between the primary producer, green algae, and the apex consumer, Chinook salmon. Ecologists have many different methods of measuring energy transfers within ecosystems. Measurement difficulty depends on the complexity of the ecosystem and how much access scientists have to observe the ecosystem. In other words, some ecosystems are more difficult to study than others, and sometimes the quantification of energy transfers has to be estimated. Other parameters are important in characterizing energy flow within an ecosystem. Net production efficiency, NPE, allows ecologists to quantify how efficiently organisms of a particular trophic level incorporate the energy they receive into biomass. It is calculated using the following formula: NPE equals net consumer productivity, assimilation, times 100. Net consumer productivity is the energy content available to the organisms of the next trophic level. Assimilation is the biomass, energy content generated per unit area of the present trophic level after accounting for the energy lost due to incomplete ingestion of food, energy used for respiration, and energy lost as waste. Incomplete ingestion refers to the fact that some consumers eat only a part of their food. For example, when a lion kills an antelope, it will eat everything except the hide and bones. The lion is missing the energy-rich bone marrow inside the bone, so the lion does not make use of all the calories its prey could provide. Thus, NPE measures how efficiently each trophic level uses and incorporates the energy from its food into biomass to fuel the next trophic level. In general, cold-blooded animals, ectotherms, such as invertebrates, fish, amphibians, and reptiles, use less of the energy they obtain for respiration and heat than warm-blooded animals, endotherms, such as birds and mammals. The extra heat generated in endotherms, although an advantage in terms of the activity of these organisms in colder environments, is a major disadvantage in terms of NPE. Therefore, many endotherms have to eat more often than ectotherms to get the energy they need for survival. In general, NPE for ectotherms is an order of magnitude, 10x, higher than for endotherms. For example, the NPE for a caterpillar eating leaves has been measured at 18%, whereas the NPE for a squirrel eating acorns may be as low as 16%. The inefficiency of energy use by warm-blooded animals has broad implications for the world's food supply. It is widely accepted that the meat industry uses large amounts of crops to feed livestock, and because the NPE is low, much of the energy from animal feed is lost. For example, it costs about $0. $1 to produce 1,000 dietary calories, kcal of corn or soybeans, but approximately $0. 19 to produce a similar number of calories growing cattle for beef consumption. The same energy content of milk from cattle is also costly, at approximately $0. $16 per 1,000 kcal. Much of this difference is due to the low NPE of cattle. Thus, there has been a growing movement worldwide to promote the consumption of non-meat and non-dairy foods so that less energy is wasted feeding animals for the meat industry. Modeling ecosystems' energy flow, ecological pyramids. The structure of ecosystems can be visualized with ecological pyramids, which were first described by the pioneering studies of Charles Elton in the 1920s. Ecological pyramids show the relative amounts of various parameters, such as number of organisms, energy, and biomass, across trophic levels. Pyramids of numbers can be either upright or inverted, depending on the ecosystem, as shown in Figure 46. 10. Typical grassland during the summer has a base of many plants, and the numbers of organisms decrease at each trophic level. However, during the summer in a temperate forest, the base of the pyramid consists of few trees compared with the number of primary consumers, mostly insects. Because trees are large, they have great photosynthetic capability and dominate other plants in this ecosystem to obtain sunlight. Even in smaller numbers, primary producers in forests are still capable of supporting other trophic levels. Another way to visualize ecosystem structure is with pyramids of biomass. This pyramid measures the amount of energy converted into living tissue at the different trophic levels. Using the Silver Springs ecosystem example, this data exhibits an upright biomass pyramid, Figure 46. 10. Whereas the pyramid from the English Channel example is inverted. The plant's primary producers of the Silver Springs ecosystem make up a large percentage of the biomass found there. However, the phytoplankton in the English Channel example make up less biomass than the primary consumers, the zooplankton. As with inverted pyramids of numbers, this inverted pyramid is not due to a lack of productivity from the primary producers, but results from the high turnover rate of the phytoplankton. The phytoplankton are consumed rapidly by the primary consumers, thus minimizing their biomass at any particular point in time. However, phytoplankton reproduce quickly, thus they are able to support the rest of the ecosystem. Pyramid ecosystem modeling can also be used to show energy flow through the trophic levels. Notice that these numbers are the same as those used in the energy flow compartment diagram in Figure 46. 8. Pyramids of energy are always upright, and an ecosystem without sufficient primary productivity cannot be supported. All types of ecological pyramids are useful for characterizing ecosystem structure. However, in the study of energy flow through the ecosystem, pyramids of energy are the most consistent and representative models of ecosystem structure. Figure 46. 10. Visual connection. Section A, biomass, indicated by dry mass G/m squared. On the left is a pyramid diagram of dry biomass in grams per meter squared in Silver Springs, Florida. The biomass of plants is 809. The biomass of primary consumers, including herbivorous insects and snails, is 37. The biomass of secondary consumer fishes is 11, and the biomass of tertiary consumer fishes is 5. Primary, secondary, and tertiary decomposers have a combined biomass of 5. On the right is a pyramid diagram of dry biomass in grams per meter squared in the English Channel. The biomass is 4 phytoplankton and 21 zooplankton. Section B, number of individuals per 0, 1 hectare. On the left is a pyramid diagram of the number of individuals per 0, 1 hectare in a summer grassland. There are 1,500,000 grass plants, 200,000 herbivorous insects, 90,000 predatory insects, and 1 bird. On the right is a pyramid diagram of organisms per 0, 1 hectare in a temperate forest. There are 200 trees, 150,000 herbivorous insects, 120,000 predatory insects, and 5 birds. Section C, energy, kcal/m²/year. In Silver Springs, Florida, the energy of plants is 20,810. The energy of primary consumers, including insects and snails, is 3,368. The energy of secondary consumer fishes is 383, and the energy of tertiary consumer fishes is 21. The energy of decomposers, including fungi and bacteria, is 5,060. Figure 46. 10 ecological pyramids depict the A, biomass, B, number of organisms, and C, energy in each trophic level. Pyramids depicting the number of organisms or biomass may be inverted, upright, or even diamond-shaped. Energy pyramids, however, are always upright. Why? Consequences of food webs, biological magnification. One of the most important environmental consequences of ecosystem dynamics is biomagnification. Biomagnification is the increasing concentration of persistent, toxic substances in organisms at each trophic level, from the primary producers to the apex consumers. Many substances have been shown to bioaccumulate, including the pesticide dichlorodiphenyltrichloroethane, DDT, which was described in the 1960s bestseller, Silent Spring, by marine biologist Rachel Carson. DDT was a commonly used pesticide before its dangers became known. In some aquatic ecosystems, organisms from each trophic level consumed many organisms of the lower level, which caused DDT to increase in birds, apex consumers, that ate fish. Thus, the birds accumulated sufficient amounts of DDT to cause fragility in their eggshells. This effect increased egg breakage during nesting and was shown to have adverse effects on these bird populations. Carson's combination of scientific knowledge and illuminating writing helped raise awareness about overall environmental issues as well as the specifics of the pesticide. The use of DDT was banned in the United States in the 1970s. Other substances that biomagnify are polychlorinated biphenyls, PCBs, which were used in coolant liquids in the United States until their use was banned in 1979, and heavy metals such as mercury, lead, and cadmium. These substances were best studied in aquatic ecosystems, where fish species at different trophic levels accumulate toxic substances brought through the ecosystem by the primary producers, as illustrated in a study performed by the National Oceanic and Atmospheric Administration, NOAA, in the Saginaw Bay of Lake Huron, Figure 46. 11. PCB concentrations increased from the ecosystem's primary producers, phytoplankton, through the different trophic levels of fish species. The apex consumer, walleye, has more than 4 times the amount of PCBs compared to phytoplankton. Also, based on results from other studies, birds that eat these fish may have PCB levels at least 1 order of magnitude higher than those found in the lake fish. The illustration is a graph that plots total PCBs in micrograms per gram of dry weight versus nitrogen-15 enrichment. It shows that PCBs become increasingly concentrated at higher trophic levels. The slope of the graph becomes increasingly steep from phytoplankton, the primary consumer, to walleye, the tertiary consumer. Figure 46. 11. This chart shows the PCB concentrations found at the various trophic levels in the Saginaw Bay ecosystem of Lake Huron. Numbers on the x-axis reflect enrichment with heavy isotopes of nitrogen, 15N, which is a marker for increasing trophic level. Notice that the fish in the higher trophic levels accumulate more PCBs than those in lower trophic levels. Credit Patricia Van Hoof, NOAA GLERL. Other concerns have been raised by the accumulation of heavy metals, such as mercury and cadmium, in certain types of seafood. The United States Environmental Protection Agency, EPA, recommends that pregnant people and young children should not consume any swordfish, shark, king mackerel, or tilefish because of their high mercury content. These individuals are advised to eat fish low in mercury: salmon, tilapia, shrimp, pollock, and catfish. Biomagnification is a good example of how ecosystem dynamics can affect our everyday lives, even influencing the food we eat. Learning objectives. By the end of this section, you will be able to do the following. Discuss the biogeochemical cycles of water: carbon, nitrogen, phosphorus, and sulfur. Explain how human activities have impacted these cycles and the potential consequences for Earth. Energy flows directionally through ecosystems, entering as sunlight or inorganic molecules for chemoautotrophs, and leaving as heat during the many transfers between trophic levels. However, the matter that makes up living organisms is conserved and recycled. The 6 most common elements associated with organic molecules: carbon, nitrogen, hydrogen, oxygen, phosphorus, and sulfur, take a variety of chemical forms and may exist for long periods in the atmosphere, on land, in water, or beneath the Earth's surface. Geologic processes, such as weathering, erosion, water drainage, and the subduction of the tectonic plates, all play a role in this recycling of materials. Because geology and chemistry have major roles in the study of this process, the recycling of inorganic matter between living organisms and their environment is called a biogeochemical cycle. Water contains hydrogen and oxygen, which is essential to all living processes. The hydrosphere is the area of the Earth where water movement and storage occurs. On or beneath the surface, water occurs in liquid or solid form in rivers, lakes, oceans, groundwater, polar ice caps, and glaciers, and it occurs as water vapor in the atmosphere. Carbon is found in all organic macromolecules and is an important constituent of fossil fuels. Nitrogen is a major component of our nucleic acids and proteins and is critical to human agriculture. Phosphorus, a major component of nucleic acid, along with nitrogen, is one of the main ingredients in artificial fertilizers used in agriculture and their associated environmental impacts on our surface water. Sulfur is critical to the 3D folding of proteins, such as in disulfide binding. The cycling of these elements is interconnected. For example, the movement of water is critical for the leaching of nitrogen and phosphate into rivers, lakes, and oceans. Furthermore, the ocean itself is a major reservoir for carbon. Thus, mineral nutrients are cycled, either rapidly or slowly, through the entire biosphere, from one living organism to another, and between the biotic and abiotic world. Link to learning. Head to this website to learn more about biogeochemical cycles. The water hydrologic cycle. Water is the basis of all living processes on Earth. When examining the stores of water on Earth, 97.5% of it is non-potable salt water, figure 46. 12. Of the remaining water, 99% is locked underground as water or as ice. Thus, less than 1% of fresh water is easily accessible from lakes and rivers. Many living things, such as plants, animals, and fungi, are dependent on that small amount of fresh surface water, a lack of which can have massive effects on ecosystem dynamics. To be successful, organisms must adapt to fluctuating water supplies. Humans, of course, have developed technologies to increase water availability, such as digging wells to harvest groundwater, storing rainwater, and using desalination to obtain drinkable water from the ocean. The pie chart shows that 97.5% of water on Earth, or 1,365,000,000 km3, is salt water. The remaining 2.5%, or 35 million kilometers cubed, is fresh water. Of the fresh water, 68.9% is frozen in glaciers or permanent snow cover. 38% is groundwater, which is soil moisture, swamp water, and permafrost. The remaining 0.3% is in lakes and rivers, figure 46. 12. Only 2.5% of water on Earth is fresh water, and less than 1% of fresh water is easily accessible to living things. Water cycling is extremely important to ecosystem dynamics. Water has a major influence on climate and thus on the environments of ecosystems. Most of the water on Earth is stored for long periods in the oceans, underground, and as ice. Figure 46. 13 illustrates the average time that an individual water molecule may spend in the Earth's major water reservoirs. Residence time is a measure of the average time an individual water molecule stays in a particular reservoir. Bars on the graph show the average residence time for water molecules in various reservoirs. The residence time for glaciers and permafrost is 1,000 to 10,000 years. The residence time for groundwater is 2 weeks to 10,000 years. The residence time for oceans and seas is 4,000 years. The residence time for lakes and reservoirs is 10 years. The residence time for swamps is 1 to 10 years. The residence time for soil moisture is 2 weeks to 1 year. The residence time for rivers is 2 weeks. The atmospheric residence time is 5 weeks. The biospheric residence time, or residence time in living organisms, is 1 week. Figure 46. 13. This graph shows the average residence time for water molecules in the Earth's water reservoirs. There are various processes that occur during the cycling of water, shown in Figure 46. 14. These processes include the following: evaporation sublimation, condensation precipitation, subsurface water flow, surface runoff snowmelt, stream flow. The water cycle is driven by the sun's energy, as it warms the oceans and other surface waters. This leads to the evaporation, water-to-water vapor, of liquid surface water, and the sublimation, ice-to-water vapor, of frozen water, which deposits large amounts of water vapor into the atmosphere. Over time, this water vapor condenses into clouds as liquid or frozen droplets, and is eventually followed by precipitation, rain or snow, which returns water to the Earth's surface. Rain eventually permeates into the ground, where it may evaporate again if it is near the surface, flow beneath the surface, or be stored for long periods. More easily observed is surface runoff, the flow of fresh water either from rain or melting ice. Runoff can then make its way through streams and lakes to the oceans or flow directly to the oceans themselves. Link to learning. Head to this website to learn more about the world's fresh water supply. Rain and surface runoff are major ways in which minerals, including carbon, nitrogen, phosphorus, and sulfur, are cycled from land to water. The environmental effects of runoff will be discussed later as these cycles are described. Illustration shows the water cycle. Water enters the atmosphere through evaporation, evapotranspiration, sublimation, and volcanic steam. Condensation in the atmosphere turns water vapor into clouds. Water from the atmosphere returns to the Earth via precipitation or desublimation. Some of this water infiltrates the ground to become groundwater. Seepage, freshwater springs, and plant uptake return some of this water to the surface. The remaining water seeps into the oceans. The remaining surface water enters streams and freshwater lakes, where it eventually enters the ocean via surface runoff. Some water also enters the ocean via underwater vents or volcanoes. Figure 46. 14 water from the land and oceans enters the atmosphere by evaporation or sublimation, where it condenses into clouds and falls as rain or snow. Precipitated water may enter freshwater bodies or infiltrate the soil. The cycle is complete when surface or groundwater re-enters the ocean. Credit: Modification of work by John M. Evans and Howard Perlman, USGS. The carbon cycle. Carbon is the second most abundant element in living organisms. Carbon is present in all organic molecules, and its role in the structure of macromolecules is of primary importance to living organisms. The carbon cycle is most easily studied as two interconnected subcycles, one dealing with rapid carbon exchange among living organisms, and the other dealing with the long-term cycling of carbon through geologic processes. The entire carbon cycle is shown in Figure 46. 15. The illustration shows the carbon cycle. Carbon enters the atmosphere as carbon dioxide gas that is released from human emissions, respiration and decomposition, and volcanic emissions. Carbon dioxide is removed from the atmosphere by marine and terrestrial photosynthesis. Carbon from the weathering of rocks and decomposition of organisms becomes soil carbon, which over time can become fossil carbon. Carbon enters the ocean from land via leaching and runoff. Uplifting of ocean sediments can return carbon to land. Figure 46. 15. Carbon dioxide gas exists in the atmosphere and is dissolved in water. Photosynthesis converts carbon dioxide gas to organic carbon, and respiration cycles the organic carbon back into carbon dioxide gas. Long-term storage of organic carbon occurs when matter from living organisms is buried deep underground and becomes fossilized. Volcanic activity and, more recently, human emissions bring this stored carbon back into the carbon cycle. Credit: Modification of work by John M. Evans and Howard Perlman, USGS. Link to learning. Click this link to read information about the United States Carbon Cycle Science Program. The biological carbon cycle. Living organisms are connected in many ways, even between ecosystems. A good example of this connection is the exchange of carbon between autotrophs and heterotrophs within and between ecosystems by way of atmospheric carbon dioxide. Carbon dioxide is the basic building block that most autotrophs use to build multicarbon, high-energy compounds, such as glucose. The energy harnessed from the sun is used by these organisms to form the covalent bonds that link carbon atoms together. These chemical bonds thereby store this energy for later use in the process of respiration. Most terrestrial autotrophs obtain their carbon dioxide directly from the atmosphere, while marine autotrophs acquire it in the dissolved form from the partial ionization of carbonic acid into bicarbonate ions, H2CO3-. However carbon dioxide is acquired, a byproduct of the process is oxygen. The photosynthetic organisms are responsible for creating the oxygen content of our atmosphere. Oxygen makes up approximately 21% of the atmosphere that we observe today. Heterotrophs and autotrophs are partners in biological carbon exchange, especially the primary consumers, largely herbivores. Heterotrophs acquire the high-energy carbon compounds from the autotrophs by consuming them and breaking them down by respiration to obtain cellular energy, such as ATP. The most efficient type of respiration, aerobic respiration, requires oxygen obtained from the atmosphere or dissolved in water. Thus, there is a constant exchange of oxygen and carbon dioxide between the autotrophs, which need the carbon, and the heterotrophs, which need the oxygen. Gas exchange through the atmosphere and water is one way that the carbon cycle connects all living organisms on Earth. The biogeochemical carbon cycle. The movement of carbon through the land, water, and air is complex, and in many cases, it occurs much more slowly geologically than as seen between living organisms. Carbon is stored for long periods in what are known as carbon reservoirs, which include the atmosphere, bodies of liquid water, mostly oceans, ocean sediment, soil, land sediments, including fossil fuels, and the Earth's interior. As stated, the atmosphere is a major reservoir of carbon in the form of carbon dioxide and is essential to the process of photosynthesis. The level of carbon dioxide in the atmosphere is greatly influenced by the reservoir of carbon in the oceans. The exchange of carbon between the atmosphere and water reservoirs influences how much carbon is found in each location, and each one affects the other reciprocally. Carbon dioxide, CO2, from the atmosphere dissolves in water and combines with water molecules to form carbonic acid, and then it ionizes to carbonate and bicarbonate ions. Figure 46. 16. In Step 1, atmospheric carbon dioxide dissolves in water. In Step 2, dissolved carbon dioxide, which is written as uppercase C uppercase O subscript 2 baseline, reacts with water, written as uppercase H subscript 2 baseline uppercase O, to form carbonic acid, which is upper H subscript 2 baseline upper C upper O subscript 3 baseline. In Step 3, carbonic acid dissociates into a proton, shown as uppercase H plus sign, and a bicarbonate ion, shown as upper H upper C upper O subscript 3 negative. In Step 4, the bicarbonate ion dissociates into another proton and a carbonate ion, shown as upper C upper O subscript 3 baseline superscript 2 negative. Figure 46. 16. Carbon dioxide reacts with water to form bicarbonate and carbonate ions. The equilibrium coefficients are such that more than 90% of the carbon in the ocean is found as bicarbonate ions. Some of these ions combine with seawater calcium to form calcium carbonate, CaCO3, a major component of marine organism shells. These organisms eventually form sediments on the ocean floor. Over geologic time, the calcium carbonate forms limestone, which comprises the largest carbon reservoir on Earth. On land, carbon is stored in soil as a result of the decomposition of living organisms, by decomposers, or from weathering of terrestrial rock and minerals. This carbon can be leached into the water reservoirs by surface runoff. Deeper underground, on land, and at sea are fossil fuels, the anaerobically decomposed remains of plants that take millions of years to form. Fossil fuels are considered a non-renewable resource because their use far exceeds their rate of formation. A non-renewable resource, such as fossil fuel, is either regenerated very slowly or not at all. Another way for carbon to enter the atmosphere is from land, including land beneath the surface of the ocean, by the eruption of volcanoes and other geothermal systems. Carbon sediments from the ocean floor are taken deep within the Earth by the process of subduction, the movement of one tectonic plate beneath another. Carbon is released as carbon dioxide when a volcano erupts or from volcanic hydrothermal vents. Humans contribute to atmospheric carbon by the burning of fossil fuels and other materials. Since the Industrial Revolution, humans have significantly increased the release of carbon and carbon compounds, which has in turn affected the climate and overall environment. Animal husbandry by humans also increases atmospheric carbon. The large numbers of land animals raised to feed the Earth's growing population results in increased carbon dioxide levels in the atmosphere due to farming practices and respiration and methane production. This is another example of how human activity indirectly affects biogeochemical cycles in a significant way. Although much of the debate about the future effects of increasing atmospheric carbon on climate change focuses on fossil fuels, scientists take natural processes, such as volcanoes and respiration, into account as they model and predict the future impact of this increase. The nitrogen cycle. Getting nitrogen into the living world is difficult. Plants and phytoplankton are not equipped to incorporate nitrogen from the atmosphere, which exists as tightly bonded, triple covalent N2, even though this molecule comprises approximately 78% of the atmosphere. Nitrogen enters the living world via free-living and symbiotic bacteria, which incorporate nitrogen into their macromolecules through nitrogen fixation, conversion of N2. Cyanobacteria live in most aquatic ecosystems where sunlight is present. They play a key role in nitrogen fixation. Cyanobacteria are able to use inorganic sources of nitrogen to fix nitrogen. Rhizobium bacteria live symbiotically in the root nodules of legumes, such as peas, beans, and peanuts, and provide them with the organic nitrogen they need. For example, gardeners often grow peas both for their produce and to naturally add nitrogen to the soil. This practice goes back to ancient times, even if the science has only been recently understood. Free-living bacteria, such as Azotobacter, are also important nitrogen fixers. Organic nitrogen is especially important to the study of ecosystem dynamics, since many ecosystem processes, such as primary production and decomposition, are limited by the available supply of nitrogen, as shown in Figure 46. 17. The nitrogen that enters living systems by nitrogen fixation is successively converted from organic nitrogen back into nitrogen gas by bacteria. This process occurs in 3 steps in terrestrial systems: aminification, nitrification, and denitrification. First, the aminification process converts nitrogenous waste from living animals or from the remains of dead animals into ammonium, NH4+, by certain bacteria and fungi. Second, the ammonium is converted to nitrites, NO2- by nitrifying bacteria, such as Nitrosomonas, through nitrification. Subsequently, nitrites are converted to nitrates, NO3- by similar organisms. Third, the process of denitrification occurs, whereby bacteria, such as Pseudomonas and Clostridium, convert the nitrates into nitrogen gas, allowing it to re-enter the atmosphere. Visual connection. This illustration shows the nitrogen cycle. Nitrogen gas from the atmosphere is fixed into organic nitrogen by nitrogen-fixing bacteria. This organic nitrogen enters terrestrial food webs, and it leaves the food webs as nitrogenous wastes in the soil. Aminification of this nitrogenous waste by bacteria and fungi in the soil converts the organic nitrogen to ammonium ion, or N H4+. Ammonium is converted to nitrite, or NO2-, then to nitrate, or NO3- by nitrifying bacteria. Denitrifying bacteria convert the nitrate back into nitrogen gas, which re-enters the atmosphere. Nitrogen from runoff and fertilizers enters the ocean, where it enters marine food webs. Some organic nitrogen falls to the ocean floor as sediment. Other organic nitrogen in the ocean is converted to nitrite and nitrate ions, which is then converted to nitrogen gas in a process analogous to the one that occurs on land. Figure 46. 17. Nitrogen enters the living world from the atmosphere via nitrogen-fixing bacteria. This nitrogen and nitrogenous waste from animals is then processed back into gaseous nitrogen by soil bacteria, which also supply terrestrial food webs with the organic nitrogen they need. Credit, modification of work by John M. Evans and Howard Perlman, USGS. Which of the following statements about the nitrogen cycle is false? Aminification converts organic nitrogenous matter from living organisms into ammonium, NH4+. Denitrification by bacteria converts nitrates, NO3- to nitrogen gas, N2. Nitrification by bacteria converts nitrates, NO3- to nitrites, NO2-. Nitrogen-fixing bacteria convert nitrogen gas, N2, into organic compounds. Human activity can release nitrogen into the environment by 2 primary means: the combustion of fossil fuels, which releases different nitrogen oxides, and by the use of artificial fertilizers in agriculture, which are then washed into lakes, streams, and rivers by surface runoff. Atmospheric nitrogen is associated with several effects on Earth's ecosystems, including the production of acid rain, as nitric acid, HNO3, and greenhouse gas, as nitrous oxide, N2O, potentially causing climate change. A major effect from fertilizer runoff is saltwater and freshwater eutrophication, a process whereby nutrient runoff causes the excess growth of microorganisms, depleting dissolved oxygen levels and killing ecosystem fauna. A similar process occurs in the marine nitrogen cycle, where the aminification, nitrification, and denitrification processes are performed by marine bacteria. Some of this nitrogen falls to the ocean floor as sediment, which can then be moved to land in geologic time by uplift of the Earth's surface and thereby incorporated into terrestrial rock. Although the movement of nitrogen from rock directly into living systems has been traditionally seen as insignificant compared with nitrogen fixed from the atmosphere. A recent study showed that this process may indeed be significant and should be included in any study of the global nitrogen cycle. 3. The phosphorus cycle. Phosphorus is an essential nutrient for living processes. It is a major component of nucleic acid and phospholipids, and, as calcium phosphate, makes up the supportive components of our bones. Phosphorus is often the limiting nutrient necessary for growth in aquatic ecosystems, figure 46. 18. Phosphorus occurs in nature as the phosphate ion Po43-. In addition to phosphate runoff as a result of human activity, natural surface runoff occurs when it is leached from phosphate-containing rock by weathering, thus sending phosphates into rivers, lakes, and the ocean. This rock has its origins in the ocean. Phosphate-containing ocean sediments form primarily from the bodies of ocean organisms and from their excretions. However, in remote regions, volcanic ash, aerosols, and mineral dust may also be significant phosphate sources. This sediment, then, is moved to land over geologic time by the uplifting of areas of the Earth's surface. Phosphorus is also reciprocally exchanged between phosphate dissolved in the ocean and marine ecosystems. The movement of phosphate from the ocean to the land and through the soil is extremely slow, with the average phosphate ion having an oceanic residence time between 20,000 and 100,000 years. The illustration shows the phosphorus cycle. Phosphate enters the atmosphere from volcanic aerosols. As this aerosol precipitates to Earth, it enters terrestrial food webs. Some of the phosphate from terrestrial food webs dissolves in streams and lakes, and the remainder enters the soil. Another source of phosphate is fertilizers. Phosphate enters the ocean via leaching and runoff, where it becomes dissolved in ocean water or enters marine food webs. Some phosphate falls to the ocean floor where it becomes sediment. If uplifting occurs, this sediment can return to land. Figure 46. 18. In nature, phosphorus exists as the phosphate ion Po43-. Weathering of rocks and volcanic activity releases phosphate into the soil, water, and air, where it becomes available to terrestrial food webs. Phosphate enters the oceans via surface runoff, groundwater flow, and river flow. Phosphate dissolved in ocean water cycles into marine food webs. Some phosphate from the marine food webs falls to the ocean floor, where it forms sediment. Credit: Modification of work by John M. Evans and Howard Perlman, USGS. As discussed in Ecology and the Biosphere, excess phosphorus and nitrogen that enters these ecosystems from fertilizer runoff and from sewage causes excessive growth of microorganisms and depletes the dissolved oxygen, which leads to the death of many ecosystem fauna, such as shellfish and finfish. This process is responsible for dead zones in lakes and at the mouths of many major rivers, figure 46. 19. World map shows areas where dead zones occur. Dead zones are present along the eastern and western shore of the United States, in the North and Mediterranean seas, and off the east coast of Asia. Figure 46. 19 dead zones occur when phosphorus and nitrogen from fertilizers cause excessive growth of microorganisms, which depletes oxygen and kills fauna. Worldwide, large dead zones are found in coastal areas of high population density. Credit: NASA Earth Observatory. As discussed earlier, a dead zone is an area within a freshwater or marine ecosystem where large areas are depleted of their normal flora and fauna. These zones can be caused by eutrophication, oil spills, dumping of toxic chemicals, and other human activities. The number of dead zones has been increasing for several years, and more than 400 of these zones were present as of 2008. One of the worst dead zones is off the coast of the United States in the Gulf of Mexico, where fertilizer runoff from the Mississippi River Basin has created a dead zone of over 8463 square miles. Phosphate and nitrate runoff from fertilizers also negatively affect several lake and bay ecosystems, including the Chesapeake Bay in the eastern United States. Everyday connection. Chesapeake Bay. Satellite image shows the Chesapeake Bay. Inset is a photo of a man holding a clump of oysters. Figure 46. 20. This A satellite image shows the Chesapeake Bay, an ecosystem affected by phosphate and nitrate runoff. A bee member of the Army Corps of Engineers holds a clump of oysters being used as a part of the oyster restoration effort in the bay. Credit A. Modification of work by NASA MODIS. Credit B. Modification of work by U.S. Army. The Chesapeake Bay has long been valued as one of the most scenic areas on Earth. It is now in distress and is recognized as a declining ecosystem. In the 1970s, the Chesapeake Bay was one of the first ecosystems to have identified dead zones, which continue to kill many fish and bottom-dwelling species, such as clams, oysters, and worms. Several species have declined in the Chesapeake Bay due to surface water runoff containing excess nutrients from artificial fertilizer used on land. The source of the fertilizers, with high nitrogen and phosphate content, is not limited to agricultural practices. There are many nearby urban areas and more than 150 rivers and streams empty into the bay that are carrying fertilizer runoff from lawns and gardens. Thus, the decline of the Chesapeake Bay is a complex issue and requires the cooperation of industry, agriculture, and everyday homeowners. Of particular interest to conservationists is the oyster population. It is estimated that more than 200,000 acres of oyster reefs existed in the bay in the 1700s, but that number has now declined to only 36,000 acres. Oyster harvesting was once a major industry for Chesapeake Bay, but it declined 88% between 1982 and 2007. This decline was due not only to fertilizer runoff and dead zones, but also to overharvesting. Oysters require a certain minimum population density because they must be in close proximity to reproduce. Human activity has altered the oyster population and locations, greatly disrupting the ecosystem. The restoration of the oyster population in the Chesapeake Bay has been ongoing for several years with mixed success. Not only do many people find oysters good to eat, but they also clean up the bay. Oysters are filter feeders, and as they eat, they clean the water around them. In the 1700s, it was estimated that it took only a few days for the oyster population to filter the entire volume of the bay. Today, with changed water conditions, it is estimated that the present population would take nearly a year to do the same job. Restoration efforts have been ongoing for several years by nonprofit organizations, such as the Chesapeake Bay Foundation. The restoration goal is to find a way to increase population density so the oysters can reproduce more efficiently. Many disease-resistant varieties, developed at the Virginia Institute of Marine Science for the College of William and Mary, are now available and have been used in the construction of experimental oyster reefs. Efforts to clean and restore the bay by Virginia and Delaware have been hampered because much of the pollution entering the bay comes from other states, which stresses the need for interstate cooperation to gain successful restoration. The new, hardy oyster strains have also spawned a new and economically viable industry, oyster aquaculture, which not only supplies oysters for food and profit, but also has the added benefit of cleaning the bay. The sulfur cycle. Sulfur is an essential element for the macromolecules of living things. As a part of the amino acid cysteine, it is involved in the formation of disulfide bonds within proteins, which help to determine their 3D folding patterns and hence their functions. As shown in Figure 4621, sulfur cycles between the oceans, land, and atmosphere. Atmospheric sulfur is found in the form of sulfur dioxide, SO2, and enters the atmosphere in three ways: from the decomposition of organic molecules, from volcanic activity and geothermal vents, and from the burning of fossil fuels by humans. This illustration shows the sulfur cycle. Sulfur enters the atmosphere as sulfur dioxide, or upper S upper O2, via human emissions, decomposition of upper H2 upper S, and volcanic eruptions. Precipitation and fallout from the atmosphere returns sulfur to the Earth, where it enters terrestrial ecosystems. Sulfur enters the oceans via runoff, where it becomes incorporated in marine ecosystems. Some marine sulfur becomes pyrite, which is trapped in sediment. If upwelling occurs, the pyrite enters the soil and is converted to soil sulfates. Figure 46. 21 sulfur dioxide from the atmosphere becomes available to terrestrial and marine ecosystems when it is dissolved in precipitation as weak sulfurous acid or when it falls directly to the earth as fallout. Weathering of rocks also makes sulfates available to terrestrial ecosystems. Decomposition of living organisms returns sulfates to the ocean, soil, and atmosphere. Credit: Modification of work by John M. Evans and Howard Perlman, USGS. On land, sulfur is deposited in 4 major ways: precipitation, direct fallout from the atmosphere, rock weathering, and geothermal vents, Figure 4621. Atmospheric sulfur is found in the form of sulfur dioxide, SO2, and as rain falls through the atmosphere, sulfur is dissolved in the form of weak sulfurous acid, H2SO3. Sulfur can also fall directly from the atmosphere in a process called fallout. Also, the weathering of sulfur-containing rocks releases sulfur into the soil. These rocks originate from ocean sediments that are moved to land by the geologic uplifting of ocean sediments. Terrestrial ecosystems can then make use of these soil sulfates, SO4-, and upon the death and decomposition of these organisms, release the sulfur back into the atmosphere as hydrogen sulfide, H2S, gas. This photo shows a white pyramid-shaped mound with gray steam escaping from it. Figure 46.22. At this sulfur vent in Lassen Volcanic National Park in northeastern California, the yellowish sulfur deposits are visible near the mouth of the vent. Sulfur enters the ocean via runoff from land, from atmospheric fallout, and from underwater geothermal vents. Some ecosystems, Figure 46.9, rely on chemoautotrophs using sulfur as a biological energy source. This sulfur then supports marine ecosystems in the form of sulfates. Human activities have played a major role in altering the balance of the global sulfur cycle. The burning of large quantities of fossil fuels, especially from coal, releases larger amounts of sulfur dioxide and hydrogen sulfide gas into the atmosphere. Acid rain is caused by rainwater falling to the ground through mostly sulfur dioxide gas, turning it into weak sulfurous acid. Acid rain damages the natural environment by lowering the pH of lakes, which kills many of the resident fauna. It also affects the man-made environment through the chemical degradation of buildings. For example, many marble monuments, such as the Lincoln Memorial in Washington, D.C., have suffered significant damage from acid rain over the years. Link to learning. Click this link to learn more about global climate change.